Welcome to the Asia Pacific seminar series today. You're being hosted by Genomics Aotearoa in New Zealand. So this webinar is coming to you from the University of Otago, which is the closest university to the South Pole, apparently, but still not that cold. It's a real deep honor for me to be here at the seminar, but also I'm not actually doing the introduction. I'm passing the introductions over to the fantastic Keiko Sakakibara, who is going to introduce our speaker for today. So thanks for coming, and over to you, Keiko.
Okay, hello, I'm Keiko Sakakibara from Rikyo University, Tokyo, Japan. Today, I'm really happy to introduce you today's speaker, Professor John Bowman from Monash University, Australia. I will introduce you his career briefly. John was born in Montana, USA, and also after John had graduated from the University of Illinois system, John got a PhD under the supervision of Aotearoa at California Institute of Technology. Now his PhD work is known as the ABC model of floral organ identity. And then he worked as a postdoc at Monash University, Australia, and worked in plant development using adaptive mutants, especially polarity mutants. And I like name of his mutants. It's quite unique and unforgettable, Yabi or Kennedy. After working as a professor at the University of California, Davis, John finally moved to Monash University as Australia. And he started a new project on the evolution of land plants using a bryophyte new model system, Marcantia polymorpha. Today, he's going to present the origin of the land flora, why all eukaryotes have homeodomain gene. Could you show your presentation, John?
So it's a great honor to be invited to give this talk. I'm in Melbourne on the lands of the Wurundjeri of the Kulin Nation. And it's also an honor for me to be introduced by Keiko, because Keiko was a postdoc in my lab in, 2010 to 13 or somewhere around there, maybe a little earlier. And the projects I'll talk about today were really started by her when she was in a postdoc. She was working on a moss called Fiscometrium, and we started working on the same genes in Marcantia while she was here. So mostly what I'll talk about is the origin of a land flora, but as a corollary, one can think about why all eukaryotes have homeodomains, right? And so in textbooks or cartoons, great moments in evolution are sort of depicted by this, with the animals looking up on shore. But the colonization of land by animals was preceded by that of plants. Otherwise, the animals would have nothing to eat, right? And so before plants came onto land, it probably looked more like this, where you have what's called cryptogamic soil crusts, and there might be cyanobacteria or other algae, chlorophyte or charophyte algae here, but there's really no soil, and it would be highly prone to erosion. So to give you guys perspective on when we're talking about, in the Cambrian period, there wasn't any plants on land, but there were algae, freshwater algae and marine algae. And then by about 470 million years ago, the first land plants evolved, and these were thought to be quite small plants. I've depicted them here as bryophytes, but that's not necessarily the case. And soil formation could actually start at this point. And by about 360 million years ago, during the Devonian, the age of fishes, one could actually walk through a forest on land that would resemble one alive today, although all the land plant groups would be quite different than they are today. All right. So there was a big Devonian explosion, and basically all the extant land plant lineages that are around today were already alive in the Devonian. So recently, with the sequencing of so many genomes, one can actually determine when genes were born relative to when attributes of land plants and algae evolved. And there's a huge number of innovations that occurred in the ancestral land plant relative to the presumed ancestral alga, which was what we would classify today as a charophyte or streptophyte alga, right? And two questions arise from this is one, why did it take so long for land plants to evolve? And why was it a singularity? Land plants really only evolved once. However, many of these lineages of algae had already evolved some somewhat terrestrial habits, meaning that the adaptation to the terrestrial environment may not have been the rate limiting step in the dominance of land plants. And our hypothesis that we're going to put forward is that the evolution of the sporophyte was really critical for the dominance of land plants. And this is where homeodomains come in, and I'll introduce those in a minute. And this, coupled with the evolution of symbiotic mycorrhizal interactions with fungi for nutrient acquisition, allowed land plants to grow large, and the sporophyte allowed them to produce lots of spores. So I'll take you through that in more detail. So, before plants and animals evolved, so maybe in the or just before the Ediacaran age, right, eukaryotes existed, but they all probably had somewhat of an ancestral life cycle. In other words, you have haploid flagellated cells that, in response to some nutrient deprivation usually, will differentiate into gametes. And this is an example from Chlamydomonas, where there's two types of gametes genetically, although they're indistinguishable morphologically. So there's a minus gamete and a plus gamete. And these gametes then fuse to form the diploid cell. And this diploid cell sort of acts as a resting stage, right? So people who work in animals often think about this style of life cycle, where you've got, after the formation of the zygote, you've got many mitoses here to form large bodies in which only a minor subset of cells undergo meiosis to produce the haploid stage of life cycle, right? But that's not the only way to become multicellular. And in the Periphycean algae, in which are related to land plants, multicellularity has evolved in the haploid stage, which means that following meiosis, these haploid, in this case zoospores, undergo mitoses to produce a complex multicellular haploid body, which then differentiates gametes. And in this case, of a species in which there's separate female and male individuals, females will differentiate an egg cell, and the males will differentiate sperm cells. And being an algae, these are motile sperm cells. They fertilize the egg cell and form the zygote. And again, the zygote forms the resting stage. And as you may have recalled or avoided recalling from your first year biology courses, land plants have what are called an alternation of generations. Since it's thought that they evolved from an ancestral charophyte algae, they probably inherited a complex multicellular haploid generation. And during the evolution of the diploid generation, they've interpolated mitoses following zygote formation. So they also have a complex multicellular diploid body. And a subset of those cells undergo meiosis again to produce the haploid spores. So just to keep people up to date with terminology, the haploid stage in land plants is often called the gametophyte because it produces the gametes. This is the haploid stage. It's going to produce the egg or the sperm cells. And the diploid stage is often called the sporophyte because following meiosis, it produces the spores. All right, so I'm sorry if I slip into gametophyte and sporophyte at times here. So one question that arises is, why would a complex diploid generation evolve? Why evolve this complex life cycle? Well, if you think about the algal ancestor, each fertilization event can produce a zygote which undergoes meiosis to produce 4 progeny. So that's not really a big problem if you're aquatic, and fertilization can happen at any time, right? So fertilization is not a rate-limiting step. However, in land plants, they've inherited this aquatic fertilization from their ancestor, and living on land, fertilization can be ephemeral. So any adaptation to allow each fertilization event to produce more than 4 progeny would be selected for. So with a really complex multicellular diploid stage here, you can get hundreds or thousands or even millions of spores per fertilization event. All right. So that's probably the major reason why, or at least people think why, a complex multicellular diploid phase evolved in land plants. And this was put forward by Bauer more than a century ago. The other thing that happened during the evolution of land plants is that in algae, it's the diploid zygote which acts as the dispersal phase, whereas in land plants, it's the postmeiotic spores that act as the dispersal agents. So each dispersed spore will have a genetically unique genotype. So I asked my daughter what she thought about land plant evolution, and this is what she came up with. I think this meant spore down here. But, you know, she thought, well, mosses and Marcantia, which we work on, are sort of down there towards the bottom of the page, whereas flowers and berries were at the top. From our perspective, we wanted to focus on these 2 major events. One is the mitotic divisions of the zygote to produce an embryo, and 2, sort of this delayed deposition of a protective coat called sporopollenin, such that the spores, rather than the zygote, act as the dispersal agent. And I'll tell you that both of these involve the activity of homeodomain genes. And as we all know from work in animals and also in plants, homeodomain genes are acting during the diploid phase of both animals and plants to pattern the body. Everybody knows about the Hox genes in flies and all other metazoans. And in flowering plants, 2 classes of tail class homeobox genes, 2 subclasses, Knox and Bell, act to pattern, in this case, the shoot meristem and the leaves. All right. But as I point out to my 3rd-year students, you know, if you look at when a gene is born during evolution, it may give you clues to its ancestral function. And if you look back to the ancestral eukaryote, it had at least 2 homeodomain genes, a non-tail class, or antennapedia class, like the Hox genes, and plants also have this class of genes. And they also have a tail class, Knox and Bell in land plants, but animals also have tail class homeodomain genes. Thus, these 2 classes of homeodomain genes must have had a function in the ancestral eukaryote, which was not patterning the fly body or patterning leaves. And it must have had a function in that life cycle, which is just a single cell, right? So some really nice work from Ursula Goodenough's lab, published almost 15 years ago, showed that in Chlamydomonas, which we mentioned briefly earlier, the minus gamete expresses a Knox protein, and a plus gamete expresses a Bell protein. And these proteins turn out to be inactive on their own, and that's because, in this case, they're cytoplasmic when they're on their own, but if they heterodimerize, when the 2 gametes fuse, the proteins can move into the nucleus and activate gene expression. And in Chlamydomonas, gene expression changes within 7 to 10 minutes after gamete fusion. So that you can think about that as a way to preload your gametes such that you can change the genetic program of that resulting zygote instantaneously, not waiting for any protein transcription or translation to occur, right? And one of the major events when Knox and Bell go into the nucleus in the zygote is it activates cell wall biosynthesis genes, so that the cell wall of the zygote is remodeled into the resting stage. And eventually downstream of that is the meiotic program, all right? So this is our model system, Marcantia polymorpha. It's basically a worldwide weed. Wherever man has gone, he is tracked around spores or vegetative propagules of this plant. So in blue here, in blue text, is the haploid phase of the life cycle. This is the vegetative thallus. It has a dorsal side that's specialized for photosynthesis. It has these little gemmy cups making vegetative propagules, which is a very useful tool in lab. On the ventral side, it has scales and two different types of rhizoids, which both anchor it to the soil and also allow some water distribution around on the surface of the thallus. It is what's called dioicous, meaning that it has separate male and female plants, and the male plants here produce motile sperm, which have to swim over using some of this capillary action with the rhizoids, in this case, to fertilize the egg cell, which is underneath these little palm tree-like things here. And each fertilization event gives you a sporophyte like this. You can see there's many, many sporophytes here. And this sporophyte is the diploid phase, and it's dependent upon the maternal gametophyte. So the maternal gametophyte nourishes this sporophyte. Another thing about Marcantia and most liverworts is that they have sex chromosomes. And so the male has a V chromosome and the female has a U chromosome, and the diploid sporophyte is a UV, and after meiosis, half the spores have the U and half the spores have the V, so a 50-50 population of males and females. So when we look for Knox genes in Marcantia polymorpha, it has basically two classes of Knox genes, and as Keiko has shown, these date back to the algae. So there's a Knox1 gene here and a Knox2 gene here. And liverworts also have two other classes of Knox related genes, but they lack the homeodomain. Right? Having a single Knox1 and a single Knox2 is the predicted ancestral condition in land plants. In Bell genes, Marcantia has three classes of Bell genes. One, which has been characterized in other land plants, which we call Bell 1 here, or actually in later slides, I'll be referring to that as Bell sporophyte because it's basically expressed during sporophyte development. And the other two classes, one is we call Bell gamete, and there's actually three paralogs of this in Marcantia. And these are called Bell gamete because they're expressed in the gamete. And the third class is something called a GLX basal. That's not our terminology, but from previous phylogenetic analyses. And nobody really knows what these genes do. And we don't know what this gene does in Marcantia either. But having these three genes, three different classes of Bell genes, is also the predicted ancestral condition in land plants. So just from a transcriptomic point of view, Tom Dierschke, who's a postdoc, who was a PhD student of the lab and now a postdoc in the lab, sorted the genes out into where they're expressed. And so in the egg cell here, we have Knox1 and some short versions of the Bell 3 and Bell 4 proteins. And then we have this, and these are the Bell gamete. And then this Bell GLX gene is expressed in the egg cell, and this is the one we don't know what it does. In contrast, these three Bell gamete genes overlapping with these are expressed in the sperms, or at least in the male side. And the Knox1 protein can heterodimerize with Bell gamete, whereas the Knox2 protein only heterodimerizes with Bell sporophyte. All right, so you get this particular heterodimer with Knox1 and Bell gamete, right, at zygote formation, presumably. And later on during sporophyte development, you get this Knox2 and Bell sporophyte forming. So we'll talk about the Knox1 and Bell gamete first, and we had a few questions. You can think about the way these genes act in Chlamydomonas, and in Chlamydomonas, the minus gamete it has a Knox protein, and the plus gamete has a Bell protein. And we have a similar situation here. So we asked whether or not Knox1 is provided by the female. Is Bell gamete provided by the male? What is the role of this Bell gamete in the female? And then the rest of these questions are for Knox2 and Bell sporophyte will return later. So first we'll look at the Knox1. And so by looking at in C2s, Knox1 transcription occurs in young egg cells, and presumably the protein's being stored there. And then later on during embryo development, it's expressed in the embryo. All right. So just to orient you, this is the egg cell here, and it's encased in a structure called the archegonium, which is a maternal gametophyte structure. And this protects the egg cell and the subsequent development of the embryo. So unfertilized, it looks like this in section. And after fertilization, this archegonium wall here develops into a structure called the calyptra. It's basically just a continuation of those cells of the archegonium wall, although they divide periclinally, so you get a multicellular structure surrounding the embryo, which is this part here. And this part here has been thought to be important to protect the developing embryo, right? It's maternal tissue surrounding the embryo. And in wild type, that embryo will eventually grow up and separate itself from the calyptra at that point, and you get a sporophyte that's producing spores in there, right? When you remove the activity of Knox1 from Marcantia, the egg cell in the unfertilized archegonium looks relatively normal. However, following fertilization, you never get anything beyond zygote development. So Knox1 is absolutely required for any post-zygotic development. Since it is expressed in this proliferative stage of the embryo, we also think it's involved in promoting cell proliferation in the young embryo, but we don't have direct evidence for that because we need to be able to have a conditional allele later in embryo development. If we look at Bell gamete, this is one of the Bell gamete genes, and it's expressed in the antheridia, which are each one of these little cuboidal cells will differentiate into a sperm cell. And just as the transcriptome data suggested, it's also expressed, it turns out, in the egg cell and later during sporophyte development. And interestingly here, in the previous slide, I didn't mention, Knox1 was only mutant in the female, and you get the 100% zygotic arrest. However, in the case of this Bell gamete, if you have a wild type female and a mutant male, you can get some embryo development. However, this is much less frequent than in wild type. In wild type, you get 80% embryos, and here you get like 10%, right? So it's not absolutely required in the male, but it definitely facilitates it. However, if you do the converse and have a mutant female and a wild type male, you see the same thing. About 10% of the embryos form that are relative to wild type. However, you can get perfectly wild type embryos. However, when you have Bell gamete mutants in both the male and the female, you approach the phenotype of the Knox1, and you never see any sporophytes develop, and you often see these multicellular entities in which the cell divisions are incorrect. Right? So this matches a lot, or very closely, what we see in Chlamydomonas, that Knox1 is supplied by the egg and Bell gamete is supplied by the sperm, and they're necessary for zygote development. The difference is that Bell gamete is also being supplied by the egg. And if one looks at the locus, sorry, I don't have a slide of that, this is sort of what we consider a full-length Bell gamete protein that's hundreds of amino acids long. And this is a short Bell gamete protein encoding the homeodomains that is expressed from a different promoter in the female. And what we don't know is how all of these interact in the zygote to promote zygote development. Right? Maybe this expression of Bell gamete in the female is a fail-safe, so that if this isn't able to kickstart zygote development, then some other condition in the egg cell allows this to start zygote development. We know it must involve other players, because if you just simply take the version of Bell gamete that's normally expressed in the male and expressed in this egg cell, you don't convert that egg cell into an embryo. It doesn't have any effect. So there must be some other physiological activity that we're missing. Whether or not that's initiated by the act of fertilization, for example, we don't know. So that's an open question. And if one thinks about the broader implications of this, in Chlamydomonas, the Knox and the Bell kickstart diploid development, and over here in land plants, it kickstarts diploid development. And if you go back to remember from looking at courses that you took about yeast, you remember the A and the alpha of mating types of yeast. Well, the A protein is a non-tail homeodomain, an ant-like homeodomain, and the alpha protein is a tail-like homeodomain protein. And so this heterodimer, the A alpha heterodimer, also kickstarts diploid gene expression in yeast, and it's also been shown in other fungi. And in fact, it's been shown in fungi and also in Dictyostelium, and recently by Mark Koch's group in brown algae, and a very recent paper on red algae. And so one can think about the reason why all eukaryotes have homeodomain genes is that probably in the ancestral eukaryote, you had two different paralogous homeodomain genes expressed in the different mating types, and that they were the genes that were required for activating zygotic gene expression or diploid gene expression, right? Interestingly, going back to animals and angiosperms, where most of the work on homeodomain genes has been done, this ancestral function has been lost in both metazoans and angiosperms. And so you only really uncover this function when you look at organisms that are often single cells or maybe retaining some ancestral function, right? We don't really know, the simplest hypothesis is that the common ancestor of all eukaryotes, a tail class and a non-tail class heterodimerized, just like what you get in fungi. But we don't really know about that, right? In metazoans, of course, you don't get gene expression in zygotes. That's delayed until later during embryogenesis. And so they have to have some other mechanism to kickstart diploid gene expression. In angiosperms, it seems that there's some sperm-supplied messenger RNA of an ant class transcription factor here that may start diploid gene expression in angiosperms. So that's the Knox1 Bell gamete story. So what about Knox2? Well, we want to know what the function of Knox2 and Bell1 is in the sporophyte. And when we knock those out, so Tom Dierschke knocked both Knox2 and Bell1 out, and they give the same phenotype, and they exhibit a maternal effect sporophyte defect. In other words, when just the mom is mutant, and you cross that with a wild type male, you get sporophytes that shrivel and die. They start out okay. This is wild type on the left, and Bell mutant in the middle, and Knox mutant on the right. They start out okay, but they fail to form a seta and elongate, and the sporophyte capsule doesn't dehisce and release the spores. Right? If you look closely, the dehiscence problem probably is due to this change in secondary cell wall modification. In wild type, the cells of the capsule wall that surround the spore sac here form these annular rings. You can see them in different views there, and that allows the cells to rip open and the capsule to dehisce. In the mutant, the annular rings instead are replaced by spiral bands, and so the spore capsule never is able to dehisce. And most of the spores, ones in green here, die. However, if you scoop those out and plate them on a new plate, a new medium, a fraction of these spores are viable, and you can maintain the line. Right? If you let them dry out, though, then they die. All right, they're desiccation intolerant. So we think that the spore coating or the coating on the spores is also affected, in addition to these change in the wall modifications in the cell wall. But you can think back to the Chlamydomonas, where one of the first targets of the Knox-Bell heterodimer was cell wall modifications, and so one can think that this may be a retention of some sort of ancestral function in that sense. To my great surprise, and Tom did this experiment multiple times before I accepted his fine work, is that Knox and Bell aren't expressed in the sporophyte as far as we can tell, but they're expressed in the calyptra surrounding the sporophyte, right? And they're only expressed starting about 1 to 2 weeks after fertilization, and then later. So they're not expressed at these very early stages, only at the later stages. So it's a maternal effect because it's the maternal gametophyte that is expressing the gene that's affecting the sporophyte. So it's non-cell autonomous. So to try to better understand this, we took NOX2 and Bell1 and expressed them in the vegetative gametophyte. And when you do that, you get target genes. In this case, this BHLH37 we think is a direct target, and orthologs of this gene in Arabidopsis are involved in expressing the tapetum and depositing kalos on the pollen. However, liverworts don't really have a dedicated tapetum. Okay. So Jonathan Levins, a PhD student of the lab, knocked out the BHLH37 gene, and turned out that it removed all secondary wall modifications in the sporophyte. So you don't see these annular rings on the capsule wall. You don't see the specialized sporopollenin deposition on the spores. In fact, they remain in tetrads. And you don't see these secondary thickenings in these cells called elaters, which are involved in spore dispersal. And when you look at the spores in detail, you can see this is a wild type spore. So here's the plasma membrane, and there's a layer called the intine, and on outside of that is a layer called the exine, which is really the sporopollenin, which protects the spores. And in other words, when you can actually find 400-million-year-old sporopollenin in fossils, this is like one of the most durable substances known to man. And in the mutant, there's very little trace of any exine being deposited. And in fact, all of these earlier cell walls that are usually degraded during sporogenesis remain there. And BHLH37 mutants have a second type of maternal effect. In other words, if you have a wild type female and a mutant male, you look basically normal. However, if you're a mutant female and a wild type male, these shrivel and they fail to expand in some cases, and it looks more like the homozygous mutant, right? So this is another maternal effect, but we think this maternal effect is happening in very different ways, right? And what's interesting is that in some cases where you have a mutant female and a wild type male, you actually get mosaics where you get patches of wild type tissue, as if this is the male allele that must be giving that, but the majority of it is mutant, suggesting that only the female allele was active. And when Jonathan looked at a number of these sporophytes, he could see that occasionally you ended up with sporophytes that almost looked wild type, suggesting that the male allele must be active. And in other cases, you got sporophytes that looked like complete mutants, which suggests that only the female or the male allele must be silenced and the female allele is mutant. And then most of the cases are intermediate. And this is consistent with this hypothesis put forward by Sean Montgomery and Fred Berger's lab, that in Marcantia, the male genome that comes in with the sperm is largely silenced throughout the entire sporophyte development. However, their data was based on transcriptomes, and they could only see a gradient, whereas here we think that it's actually individual cells that the male genome becomes active or does not become active, all right? And you can think about that in terms of imprinting, because the sporophyte is dependent upon that female. It's a placental arrangement, right? Furthermore, we could find some plants or sporophytes in which the spore mother cell wall that's encasing the spores is still present, and yet the elaters are wild type. And this strongly suggests to us that the actual tapetum, or the wall of the sporophyte, is acting as the tapetum to deposit to degrade the spore mother cell wall and to deposit the spore pollen on the spores. And so in summary, this would suggest that this heterodimer is active out here in this gametophytic tissue and is acting non-cell autonomously to activate the sporophyte, and that BHLH37 is active in the sporogon wall here, the sporophyte wall, that's acting non-cell autonomously to put the exine and some other things on the developing the meiotic spores, all right? And this is interesting from a very broad perspective, in that if you look at the fossil record of land plants, at about 465 million years ago, you start getting these tetrahedral tetrads, which have been interpreted. This is like 400 million year old sporopollen in here. These have been interpreted as being embryophyte or land plant spores. Before that, you get a bunch of random, or not random, but spores that are not produced in tetrads, right? However, the first macrofossil record of land plants isn't for some time later. And so one idea is that there may have been a gradual acquisition of land plants are otherwise known as embryophytes, of land plant characters here. And one idea that we had recently is that, we always draw a land plant life cycle where you get a zygote, and then that zygote produces a little embryo inside of the calyptra there, and that differentiates into these foot and seta and sporodinous tissue. However, it could be possible that early land plants, very early land plants, this whole group of cells here all underwent meiosis, and then you'd get this little bag of spores. And if the calyptra could act as a tapetum to deposit the appropriate sporopollen and other things onto the meiotic products there, then this would be a stage in the fossil record where you basically see spores, but not much else in terms of the sporophyte. Okay, so how much more time do I have? I think I'm running out, eh?
Almost.
So I'll skip the evolution of sex chromosomes, but I'm happy to take any questions on that. And the work on the Knox1 and the Knox2 in Marcantia was all done by Tom, and Jonathan did all the BHLH37 stuff. And I'm happy to take any questions.
Thank you, John, for wonderful talk.